This is your Module 1 Basic Information video, and the whole point of the video is for people who couldn't make the meeting or you are struggling on your own at home to do your Clostridium work to have a place to go. So first of all, the first thing you will need is the training manual. Now if you came to the meeting, I had a copy for you already printed off. You can come see me for one if that's easier for you. If you're trying to work right now though, there's a way to get there, so I'm going to refresh you on it one last time. You go here to your Office 365. You always make sure you pick the work or school account with the little ID badge and enter your current password. So once you're in, you might land at your email. If you want to change that, by the way, you come up here to the gear, you come down to App Settings, Office 365. And when you get here, the settings should be highlighted and it's going to give you an option for Start Page. I have mine set to OneDrive. If you click here, it'll let you go all sorts of places. Okay, you can pick whichever one you want. So if you wanted to know how I changed that, that's the easy one. Make it log into the page you want. To get to our stuff though, you'll click the waffle, you'll click people because you're looking for our group of people. And once again, yours is going to look different. Yours might not have anything in the middle, but we are the Bioinformatics Experimental Group. So you'll click that group and our resources will pop up, including again, people in here. If you got to contact somebody in the group for help, you can email one of us. You can go to our calendar to see when the next meeting is. And the place you would go is the files. The notebook and connectors I don't use. But if you click Files, you'll once again get to that place where there are a few resources for you, most notably the training manual. So you'll want to pop that open almost every time when you're working. The rest of this stuff is not necessarily required. Training manual, really important. And since this video is about Module 1, you're going to just download Module 1. Clicks and downloads. And it'll open in a PDF, so anybody can get it. So we're going to kind of go through this module together. I am not going to read the book to you. I am just going to try to do the process that's in the book. So I'm not going to flip back and forth, okay? I'm just going to summarize for those of you who couldn't make the meeting. So what you will need for any kind of GENI-ACT work would be a browser. They recommend Chrome and Firefox the most highly. I would not use your Internet Explorer. So here I am. I happen to be using Firefox, and it has always worked for me without a problem. So I can definitely recommend it. The school does let you download it. You can download it at home. It is free. It's just a browser. You guys all know about Chrome. It is GENIACT.org. The manual says .com. It is .org. You have all signed in here already. Okay, but Dr. Corey from UB has been in here since then, and he has assigned you your gene number. So you are ready to go. So what you'll do is you will click login. And I have mine set to remember and my password. I would recommend you do the same, or that you write it down on something or make a contact in your phone. But this thing is definitely a thing that you would want to favorite this page, because then you're going to be coming back to this online GENI-ACT notebook all the time. So just favorite it and you'll be able to get here very quickly and efficiently without having to remember the URL and type in your login and password all the time. So anyway, when you enter it correctly, I asked you to use your school email for your username. If you picked a different one, that's up to you, but try it and then you'll click login. Once you're on the landing page, you will see some just very, very basic information where you can control your own password, okay? But yours will say something different. This for me is the training program that I went through to be able to teach you how to do this. You will see something like this: Benedict, Clostridium botulinum, SEPA Year 2. Click it and you'll see some basic things up on the top. I see something different. I see all of you. So at any time I can get into your notebook. If you need to send me an email and say, can you check this part of my notebook? I don't know what I'm doing. I'm struggling and I need help. I might be able to help you without being right with you, which is kind of cool. Or I might say, come see me. But I opened up Jared Goodings, okay? And he has been assigned this gene that has been called by the computer. And the computer basically thinks there's a pretty good chance that there's a gene there, but it doesn't really know much else about that. So what Jared would see is that he has a notebook. You don't have to memorize these numbers. That's for his Clostridium gene. There's also a notebook for Chytococcus sedentarius 00010. This is the one that we are all going to start with and practice. I am going to go back into my notebook, so I'm not messing with Jared's stuff. So this is my notebook that I used when I annotated to practice this gene with KSED 00010. This is just the the abbreviation. This is the practice gene. So this is my lab notebook. I'm not messing with any of your stuff. So I'm going to open it. And one of the things you're going to see if you scroll is it has some instructions up here. It'll pop instructions up. But then the rest of it is very simple. It's just a notebook. So what you would do is you'd click the plus and it'll open some things up. Our goal for this first video would be just to complete the basic information about our bacteria. So again, using KSED as an example, you'll see mine filled out already, but I will walk you through it. Okay, so first you'd click the plus to expand this section of the notebook. These are like binder tabs. So let's go into the basic information section. You'll see that I have stuff already filled out here. My goal is just to show you how I did it, okay? And it'll be pretty basic. So it says DNA coordinates, go to the gene page. And one of the best things that I can tell you to do and get in the habit of doing is opening things in a new tab. So what you do is you press Control key and you click this link. And you can see it automatically made a new tab pop up, and I'm going to go there. And KSED 00010 is going to be the yellow gene. When you do your Clostridium one, that your gene will be in yellow. So once I'm seeing this yellow thing, it tells you in your manual your gene is yellow along with other genes in the neighborhood. So these other blue things might be genes that some other people in our group are working on. In this case, they're KSED genes, so these are just samples. And because our arrow in this sample is pointing to the right, it means that the gene is on the top strand, or the plus strand. If your gene is pointing to the left, like these guys, that means it's on the minus strand. So we're going to need that information in a second, okay? But for now, we're going to drop down, and it says that the coordinates are 209.1729. These are base numbers, DNA base numbers. It's base number 209 and it extends, the gene extends until base number 1729. That's a lot of letters. So you're going to highlight that. Control C is how you copy really quickly, or you can right click and copy. You go back to your notebook and look at this. I already have it there. You'll click this little notebook icon, and what I did is I copied, I deleted the second number, and then I pasted, which is Control V, and I deleted the first number. Okay, and because it is going to the right, it's going to be the forward strand. You have to click save in this notebook. It's very simple and easy to use, but if you were to say cancel, it would take you back out and you would have to do it again. So we want to press save, and that part's done, believe it or not. So the next part, the DNA sequence. You're going to go back to that other tab where we had the gene page. Now you could do it again. If you shut the tab by accident, I'll do it. Oops, I shut it. I thought I was done. Okay, crap. Control, click it, and it opens the tab again. No problem, it is the same thing. So we just did the coordinates. It does tell the length, I'll need that in a minute. The length of the gene in nucleotides, but then also in amino acids. It has a name that the computer's proposed, along with the idea of what that might do. So we're gonna skip beyond that till it says nucleotide sequence. Back in my notebook, I need the nucleotide sequence, and you can see I already put that in. So I'm gonna go there. Notice that it's grayed out at the beginning and at the end. So I'm going to highlight only the black letters. It's highlighted. I'm going to Control C or right click copy. It's up to you what you want to do. Go back to your notebook. Remember, it won't let you do anything until you click this little icon. And when you do, I will go ahead and delete what I have quick. You will Control V or right click paste. Now your manual will tell you that that's not quite good enough. We want to make sure that we have what's called a FASTA header. We will be using this nucleotide sequence later, and it says right up here you have to use FASTA format. So what you're going to do is go back to the beginning, and you're going to press enter to kick everything down a line, go back up, and when you do this symbol, you'll have to find it. It's really the greater than symbol. What it's going to tell the computer to do, or all these programs we're going to use, is to ignore that line. So what we're going to do is say this is, well, Chaetococcus sedentarius, and then we're going to use the gene number 00010. That's the name of the gene, and we're going to say what this is. So the computer, or any program we use, is going to ignore this first line, but we will always know what is this jumble of letters that we're messing with. It kind of helps you to keep track of what's going on. So then when you're done, you press save. When you're doing your Clostridium bacteria, you're going to do the same thing. You'll paste its unique nucleotide sequence, but then you'll do the little caret sign and you'll say Clostridium botulinum and the gene number of the gene that you have. Let's say you forget. Come back up to the top and it gives you the locus tag. So I could literally copy and paste this in. You will eventually get to know your number very well. So far I did it. I have it FASTA formatted, which is important for the future, and I make sure when in doubt, I'm clicking the save button. I don't want to lose any work in here. I don't need to upload an image at this point. There's no image. I copied and pasted. There will be a time in the future where you'll need to. The last part is about sequence length. So I go back to my other tab. I'm going to look for the box that's labeled length. So on my other tab, it would look like this at the top with that yellow caret. It says the length box, and I told you we'd be coming back to that. So I'm just going to copy that for now. Remember, I told you the first number is how long the nucleotides are, how many letters in the DNA there are, ATCG. The second number is what that means in terms of amino acids, how many there are. So I'm going to copy that. Go back to my notebook. In this notebook, in a science notebook, too much information is better than looking back later and, wait, what did that mean that I wrote? That's called a waste of your time. So include enough information, click save. Scrolling down from there, I need a protein sequence. I already have the gene page open in my other window. So I scroll back down and I saw the nucleotide sequence is right there. We already dealt with that. So I'm going to go to the amino acid sequence, and it's much shorter because remember, 3 of these letters will code for 1 amino acid. So this should be about 1/3 as long as that, and it looks pretty good. So I'm going to copy. I'm going to Control C this time because it's so much faster. Go back to my notebook. I will erase what I have here. This is what your notebook would look like. And I'm going to paste it in. Go up to the top, and I need that FASTA header again, the greater than sign, and I'm just going to write what this is. K said 00010, and in this case, it's the amino acid sequence with a colon. Computer will ignore that first line because it's FASTA formatted, which is what we want next time we meet. We will use this stuff next time we meet. And then I click save. Once again, there's not a picture to upload. And lastly, the sequence length. This is actually that same number from up here. So I will come up and grab it. I copied it, Control C, and I actually am going to leave that just nucleotides because it's cleaner. And down at the bottom, sequence length, 506 amino acids, save. And believe it or not, I'm at the end of Module 1 already. All we've done is gotten our gene into one place, and we've established how many amino acids there are. Very basic information. You should now go and do the same thing with your Clostridium bacteria. Always make sure you save, and when you're done, make sure you log out. Okay, so at the end, if it was me, I would do this. I would scroll back up. I'd click the minus, I would open up the next thing. I've already done stuff there. I'd go back in, and I would make sure, yep, filled in. That's filled in. Filled in, filled in, and filled in. I'm done with that gene. So when you're ready for your Clostridium gene, this is a breadcrumb trail. You can click back to courses or to Benedict Clostridium and find your actual bacteria this time, and then dive in. Make sure you save, do well, and next time we get into sequence-based similarity, where we'll look at what these sequences mean and start annotating our gene. Thanks.